our next speaker is Yinhai Wang from AstraZeneca. And Yinhai is going to talk about opportunities and challenges in multimodal, multidimensional image analysis for drug discovery.
Thank you very much, Carla. And okay, so I'm Yinhai, I'm from AstraZeneca. So slightly different kind of a talk, and so I'm going to talk about rather than results of experiments we have done. It's more like something fairly new to me and to our company. So first of all, to introduce where I'm coming from, so I'm from AstraZeneca, and there's different departments, different sections in the company, and so there's many different disease areas. For example, the cardiovascular disease, and there's oncology, and there's respirational disease, and besides that, there's another department called discovery sciences. So basically, in the department, we're actually dealing with different technology, from biology to chemistry to people like us. We're dealing with computer work. So within discovery sciences, it's our department, so-called quantitative biology. And within that, we have 4 different branches. There's bioinformatics, and there's mathematical modeling, imaging, and statistics. And so our whole department of the quantitative biology will have around 30 people. So we all have this kind of mathematics or computer science background. So kind of group of nerdy people, and I'm lucky to do imaging because people know what I'm doing, what imaging is. And if you imagine somebody actually in our group try to do mathematical modeling, try to explain that, what is that to a biologist? So it's tricky enough. But anyway, so today I'm going to talk about something so-called mass spectrometry imaging, or short for, you can shorten it to be mass spec imaging, or MSI. So first of all, I'll give you a quick look at what the image looks like. So the image on the left-hand side is actually what we obtained, so-called MSI image. It's fairly low resolution, and so it could be a couple hundred by couple hundred pixels in size. And on the right-hand side, basically, it's like you can put on kind of pseudo color, some heat map on top of the left-hand side image, then give the image a bit color. So what does that actually come from? So what is mass spec imaging? So if you think about it, you got an animal, and then you take the brain out, and then you fresh the frozen sample, and then cut a single slice. So you got a slice of a tissue, and then you run that piece of fresh frozen tissue into a mass spec imaging machine. Basically, you can think of that like a big black box. And what happens, like for every single point in that fresh frozen tissue, it's corresponding to a single location. Corresponding to a whole spectrum of signals. At a single location, that location is corresponding to a certain amplitude. That amplitude can be translated into a pixel value to be either high or low. So the image on the right-hand side, which is an MSI image, is corresponding to the similar shape to the fresh frozen sample. But however, not only at this spectral location, but there are actually many different spectral locations, which is like these. So at different spectral location, you are getting a slightly different or very different imageries. So in terms of the image itself, the mass spec imaging is actually a whole stack of many thousands of images. And each single one of the image can be fairly low resolution, a couple hundred pixels by couple hundred pixels in size. But we also have the kind of newer version of the mass spec imaging, which is actually fairly high resolution. I got the impressions like something like its resolution is about 5 micrometer. And so that is case of what is actually the meaning of actually at the different spectral location. So mass spec imaging is able to detect and visualize the chemical composition of a certain sample. Slightly different type of imaging. So it's actually detect what's actually in you, what's the chemical how the chemical actually inside a piece of living tissue. And so if you think about it, that's actually the case. So that for that it is able to detect different things. For example, it can detect the what's actually the intrinsic metabolite inside a animal or human. So it could be a amino acid, could be fat, or could be some lipids. So whatever it is, they're all chemical. So something basically intrinsic to you yourself, or could be a certain drug. So if I'm being injected to a certain drug, the drug itself is a certain chemical. So the drug goes to somewhere in my body, and then I actually visualize that. Or it could be a biomarker. So you have already heard a lot of talk today about the biomarkers, and I think maybe the easier way to explain it could be a biomarker could be a kind of indicator to a certain disease, or if the patient is going to respond to a certain drug or not, or to maybe other measurements. So basically, that's also can be a kind of a chemical abundance inside you as well. So by thinking that way, the MSI is actually able to describe the intrinsic metabolite and drug concentration, and also the biomarker simultaneously in a piece of tissue. So because of that, so we think that actually MSI could be used to investigate the drug's efficacy and safety. So we want to try to use that to investigate if a drug is actually efficient enough, or if a drug is actually working, or if there are going to be any toxic side effects to the animal or to the human. And first thing which is worth mentioning, which is MSI is actually label-free. So basically, you don't actually stain the tissue with whatever chemical, which may actually you may actually damage the sample itself. So without any labeling, so we just get the fresh frozen tissue, then you get the images. So that's more likely to be the original look of what you're going to get. Basically, another example. So basically, at different spectral locations, you get a whole set of thousands of images, and each one of them could be looking very different. And some of these, like even the external boundary is kind of missing, and so could be so it could be fairly different, or maybe similar for some of them. And then the MS actually doesn't stop at the image itself. The whole set of MSI images can be actually working together with the standard hematoxin in the ocean stained imagery, or short for HNE images, which is the kind of a gold standard for pathology over the last 100 years. So pathologists use a piece of tissue stained with a hematoxin, kind of a blue and pink colored staining. Then basically, they use that and put on a piece of small glass under a microscope. Then they can view to see what's actually the tissue structure and the morphology of a piece of tissue. So that's actually being used as a gold standard for diagnosis and prognosis of different pathology studies. So based on this, so if you think about it, you get a kind of an organ, let's say it's a brain, you cut into half. So then you get one slice from here to do the MSI image. Then from another side, you actually have another slice to do the H&E staining. So this actually two slides, actually they're neighboring slides, so their morphology or their chemical composition is very similar. So you get a very close relationship between the MSI image and the hematoxin image. Hematoxin in the ocean image. So this is just a kind of blow-up image of a H&E slide, and so pathologists look at these and think about, okay, this is a piece of lung tissue, and the pathologists think, okay, that's a bit, quite big of a chunk of a tumor going on here. So it's a squamous carcinoma, and so this is all the cancer cells, and here's with some dead tissue, so-called necrosis, and here's probably all right. And then pathologists think, oh, that's probably, by I look at the image itself, I think maybe there are 70% of tumor cells. And so for the last 10, 20 years, so there's a kind of an emerging market or a scientific discipline, so-called digital pathology. So we're able to actually use a computer to analyze this kind of pathology images, and that's actually thankful to the development of this kind of glass scanners. So a single piece of slide, but that size, can be scanned into ultra-large digital images at very high magnification. So that image itself could be 200,000 pixels by 200,000 pixels in size. So after JPEG compression, you're talking about that's probably, I don't know, 100 gig or something like that. So it's a fairly large image, but however, with availability of this digital imageries, and then we can actually apply lots of different computer algorithms to detect different compartments to do machine learning. For example, in this case, we use a machine learning method to find the boundaries of where the actually tumor is, and then we use the heat map to show the concentration of the tumor cells. So, because we can actually make H&E images and MS image working together, then actually open up a huge opportunity to study many different things. For example, if we are going to get one MSI image and one H&E image, so MSI there could be a certain metabolite you're actually interested. Then you want to study the relationship between the metabolite itself with the tissue, or maybe subsections of tissue, and you can certainly do that. And but however, then it's a case of how do you actually simple things like how do you visualize that, and how do you gonna do it, and that's a question itself. Or you can actually study a certain drug. You want to say, okay, where is this drug actually located on my tissue, and why is that drug is only in that area, but however not in my interest areas, and what's the regional difference between this region I selected and that region I selected, and so you can actually study a certain drug. And also you can study the drug concentration at different locations, and also the spatial relationship between the drug and tissue morphology. Similarly, you can study the kind of interrelationship between the tissue itself and also certain biomarkers you're actually interested. And so basically, long story short, so with the ability of MSI images, so we are able to actually combine all of these different metabolite, drug, and biomarkers together, and together with the H&E slide imagery, so that we can actually study all of them together. And it comes with a number of problems. For example, registration is a problem because it's cross-modality registration. Between MSI images, they are very good, well aligned, so you don't have to do registration at all. But however, you need to register the H&E with MSI image. And it's a case of if you use a stack of MSI images, how do you visualize something you're interested? It's very simple questions, and if you cannot see it, and how do you explain it to your colleagues? And the tissue image processing, and so which is it's like, for example, to use the computer algorithms to recognize tumor or different compartments in the tissue. And also there's a number of multiple dimensional data analysis problems because you're using multiple MS images, and MS images have a certain intensity which indicates something, and also it's the spatial locations and the distribution of the information. And also the tissue structure itself from H&E is complex enough, etc., etc. And then makes things a bit worse, and I won't say worse, but a bit more complex. There's another type of technology which is widely used in molecular biology research, so-called immunophysicochemistry, or IHC. So what you see here is a piece of breast tissue. So there's two colors in it. There's a blue color and this brown color. Blue color is also hematoxylin, it's kind of a chemical dye. The brown color is let's say in this case it's called HER2. HER2 is a biomarker, which is HER2 actually can identify if a patient has breast cancer or not. If there's no brown color in this image at all, which means there's no HER2, but however, if there's a very strong brown color in this case, which is, and you can see it's kind of like every single cell membrane is actually surrounded by very clear brown color, which means this patient has a very strong HER2 expression, and so which means that she has this kind of a type of breast cancer, which can be treated by a certain chemical called a certain drug called Herceptin. Then she will be respond to that kind of chemical, and basically her tumor would be likely to be controlled by the drug itself. And there's another type of imagery, which is you also know, I think it's fluorescent multiplexing. Basically, you are able to stain a piece of tissue with multiple different biomarkers in fluorescent channels. In this case, you have 1, 2, and 3. This one's kind of a cell nucleus marker, and this one's probably a cell cytoplasm marker. So nucleus is inside, it's kind of solid, look like a solid ball shaped, object inside the cell. The cytoplasm is actually the region surrounding the cell nucleus, so they look like slightly bigger than the cell nucleus, and actually you can actually overlap all of them together to be one of these, so it become a color image in a way. So by having all of these, so now we have basically we have the capability, we have the tools, we have the technology. Actually, we can produce all of this together, which is you can stain. So if you have a piece of tissue, you cut many different slices, and you're interested in a number of biomarker in HCs, you can cut many slices, and you can use the next slice to stain with H&E, which is to study the cell morphology. This is to study certain biomarkers, and you can have a whole stack of MSI images as well. And also, if you want, you can also have a number of slices to do fluorescent multiplexing as well. So basically it means that at this stage, we're actually dealing with a multimodality of these 4 different modalities. Each one of them contains a great number of imageries. So we are able to study HC plus H&E plus MSI plus fluorescent all together. And in a kind of a not really strict way of saying this, so we can say we are able to study the protein biomarker expression plus tissue morphology plus the metabolite and drug concentration and biomarkers as well, and also study the DNA signaling. So all of these can be studied together. So if you think about it, it's like if you only just take 2 slices of this image out, and that's already something. For example, in this case, so you're actually you're studying maybe 2 HC stained slides. So one of them is like underneath brown colored image. So certain brown color, certain biomarkers actually expressed in here, but however the second biomarker actually expressed outside, it's kind of a brown region outside, which I use the kind of heat map to actually show that. So that actually shows you the kind of interrelationship between the 2 kind of biomarkers. So there's endless combination you can actually do, endless possibilities, and I'm actually a bit scared in terms of we are able to do this, but however, we don't really have much methodology and much tools actually to do this properly. And a scientist in our company could actually knock on my door tomorrow to say, Yinhai, I got all these images, would you do some image analysis for me? And I to be honest, I don't really know how to do it really properly. So basically, long story short, so in this slide we say that there's multiple image modalities for HC, H&E, MSI, and fluorescent. Each one of them contains a number of imageries. So we're hitting the kind of problem of multimodality. And it makes things worse, and you can actually use MSI to study those responses. So for example, for a normal mouse, for this guy, this is a kind of his some metabolite he has in terms of in his brain. So there's the glutamate and aspartate and glutamine. So that's what the normal look like. He hasn't been treated with any drug at all. But however, if you give him if you give a second mouse some drug, then the image, the MS image actually changed for this guy. But if you give a third mouse a double dosage of this certain drug, the image also changes as well. So in our company, we would actually be interested to study the kind of dose response in terms of if you give an animal or human a different dosage of certain drugs, and what actually changed, what actually preserved, and blah blah. So there's basically this kind of an extra dimension of data in terms of dose response, and it's also worth to mention that the image registration in this case could become a problem simply because this image and that image are from 2 different animals, so they're not from the same person or same mouse. And we don't have a real example, but however, it can also be a case of so you actually treat a certain sample at a given time, and then you actually could be treating the same sample with the same drug, but however, for a longer time, then the image actually looks different. So basically, using these, actually, we're looking at the kind of a change in terms of the time scale. So that's another dimension we have to actually think about. And finally, I want to mention this kind of so-called Swedish Farad model. And so what it is, so the image on the left-hand side is a Swedish Farad at a very macro level, so it's very small. The best way I can actually describe that would be something like the right-hand side, which is a sticky rice. So it is more like that. So lots of cells actually stick up together to form a kind of 3D structure. And the reason why we actually use the 3D spiral model simply because, as you probably already know, so almost 90% of early clinical trial for a novel drug fails, really high failing rate, and simply because if you think about it, you try to do some drug development and you try to grow out some cells on a flat dish, the interaction actually disappeared. So that's why we think maybe it's like we should actually somehow form a kind of 3D structure so that cells have some interaction to each other. We're trying to develop kind of novel model platform so that it's more likely to simulate what the kind of human environment actually is. So it's actually a fairly topical area to study kind of novel preclinical model platforms. So the reason why I want to introduce that simply because there's actually a published paper and people actually try to use MSI for the 3D spheroid. So they grow this kind of 3D spheroid in the container. Then afterwards they treat it with certain drugs. Then afterwards you got the kind of a treated 3D spheroid like a ball, like a sticky rice ball. Then you cut it into many different slices. Then afterwards you run into MSI, and then you got a whole stack of images per slice. Then you got every single slice, you can actually get whole stack of images. So basically you are getting, end of the day, you're getting lots of different 3D balls. Then you compare all of them. So which means it's like we are actually getting another dimension added in here. So basically, this is the kind of problem we're facing at the moment. So we are dealing with the availability of MS images, we're dealing with multiple image modalities, including immunohistochemistry, hematology, MSI, and also fluorescent. And also we're dealing with multiple image dimensions from standard 2D images and those different color channels and from different staining, and also the dimension of dose response, dimension of the time sequence whenever we treat the animal or human with the same drug of different time points. And then you have the kind of 3D structures. So it's like it really is it is a multi-dimensional data problem. So I introduced all of these. So this is where we are at the moment. After spending so much money and so much data being generated, and after we have done a long period of time of work, this is what we got. A tiny Tony plot sitting at the corner of an A4 page on the paper in a mediocre publication. It's a pity, but that's what happened. Simply because I think many reasons to cause this. It's like one way is like we can think about. So like we haven't done a really good enough job to educate our colleagues about the real power of this new technology. What this can actually do. This actually can be working together with different modalities, and we can actually analyze the data in different dimensions. So that kind of conversation hasn't been in place, and also because people are actually specialized in different domains. So it's hard for one person maybe specialized in PCR to understand what is actually immunohistochemistry. So it's many different reasons. So we're actually not there yet, but this is just the reality. So actually, with all of this technology together, we can study so many different things, and some of them actually fairly easy, some of them can be quite difficult, and they are all very topical research areas in molecular biology and pharmacology, and they have very big impact for drug development. So, and also we think it's like maybe machine learning could actually help, and simply because MSI is a stack of images, and the matching between the tissue morphology with MSI is could be probably used the machine learning, or maybe the matching between a HT expression with MSI. For example, if we want to find out from a stack of MSI images which one actually most similar to a HER2 expression in the protein domain, or maybe a combination of a different spectrum together, etc. But however, we also expect machine learning, actually we would expect machine learning to play a very important role in MS images, and the following area I think is the area we should actually pay attention, and we should allow the machine learning to take into human input, expert input, and also maybe an element of active learning. So if we got some classification result, and we should allow the domain expert to actually retune the result and give feedback to the training itself. And simply because we're dealing with a lot of uncertainties, and we're dealing with unknown chemicals, with unknown cell lines, and so we have before we do it, we don't know what the kind of chemical would actually do to the sample itself, so we just don't know. And so in summary, in AstraZeneca, we're investigating novel imaging modalities all the time, and we're generating a large amount of multimodal imagery data, and in turn, a large amount of multidimensional numerical data. We would like to pursue better image and data analysis method, machine learning method, so that we can actually unleash the power of this multimodality and also multidimensional data. And finally, to do all of this, our final goal is actually to help our colleagues and help myself to understand the molecular biology better, to understand the drugs and mechanism of action better. So I'd like to thank you all my colleagues at AstraZeneca, and also our collaborators in the Netherlands. And certainly I'd like to thank John and Carla for having me here. So thank you very much.
Thanks a lot, Yinhai. Other questions?
I can make a comment. We actually developed cars and Raman imaging for quantitative imaging analysis for our colleagues at Cancer Research UK. And all we got was a little plot like you showed. So maybe we should talk and work together and educating our colleagues.
Yeah, sure, absolutely.
I think sometimes it's quite hard when you develop something new that people actually just don't know what to do with it. And
absolutely, yes.
It actually has to come together. So, but that's actually quite hard.
Yeah, exactly. That's absolutely right. Yeah.
But the MSI imaging is very interesting. It's very. What is the resolution you can get?
I think the resolution, the higher resolution version we can get to is a 5 micrometer, which is a fairly high-ish, not as good as digital pathology, but it's a fairly clear. But quite slow though, so the technology is still under development, and so generally the image is actually fairly slow. That's just what it is.
What is the sensitivity of the metabolites or drugs or anything?
I have no idea, to be honest. I think probably I was the wrong person, and I stopped to do chemistry after I was a 16-year-old, and I don't know. Sorry.
Other questions?
I will just comment. Thank you for summarising what the pharmaceutical industry likes in terms of decision making, much more eloquent than I did this morning, by just showing you a little bar chart with a small P number, which is, after all, I think what everybody, after you get past a certain level of seniority in a pharmaceutical company, that's what everybody wants. I think there's a recurring theme there, which is about plausibility and trying to convince people that some of the complicated methods that are getting ever more complicated are actually useful and aren't just something to be feared as a black box. Have you got any thoughts on how we achieve that?
I got the kind of tendency of feeling, which I'm not sure if I'm doing the right thing, which is I'm trying to use simpler methods for the reasons of, for example, if I need to do a classification, I probably use decision tree, simply because I can explain that to a biologist very easily. People know what the tree is, rather than have to explain a black box with a SVM inside. And I can explain some features fairly easily. For example, what is the brightness of a certain object, what is the shape of an object, which is fairly simple. If I'm trying to explain a more abstract feature, for example, like texture or entropy, to a biologist, I would have trouble. So if they don't understand me, they won't trust my result, they won't trust my methodology, then I should run into trouble. So which doesn't mean the methodology itself have flaws. It's more like I cannot explain it to the person, and then they won't use it. So which means the methodology I'm going to develop is less practical, although it has a better result. But however, I cannot prove it to them. So
yeah, absolutely right. It's a very important point for us all to think about. Thank you.
Thank you very much.
Okay, thanks a lot.